it is not able to do the scale to the kg or ton level, doesn't mean anything to the company or the professionals. Then now I have developed the lab scale product. But how I will method validation. So all this portion is part of our development into the R&D, where organic synthesis plays a critical role. Now, once I've done the development, I've done the method validation, now it becomes the optimization. Because one batch I prepared, it's okay. Second batch I prepared, something went wrong. Third batch I prepared, something is very different wrong. That is why it's called as a process optimization. So whether whatever the product or the process I develop, whether it is optimized process, that we need to do into the lab before we go to the larger scale. So once the optimization is done, then we need to go to the next stage. It is called as a pilot batch manufacturing. So 100 gram, now we will go to the 1 kg. Many companies, they do the mistake from 100 gram, they go to the direct to the 100 kg and they end up with many issues. So my suggestion to industries and my suggestion to all the students, that when we do the, as I mentioned, we have to go step by step. So here also is a step, like from the left to the pilot to the full scale. So pilot batch manufacturing, that is for the reproducibility and confirm the process at that level. Then, now the another thing in the pharma is called as a scale up. 100 gram to 1 to 100. So there is a scale up. So in that scale up, the reactors, the processes, equipment, people, everything is getting changed. So once something is getting changed, there are always a risk which is associated. So in the recent pharmaceutical norms into the R&D, into the manufacturing of the API, where organic synthesis is playing major role, that where the risks are associated, like whether risk is into the chemistry, whether risk is to the equipment design, whether risk is to the selection of the raw material, whether risk is into the vendor. So all these are the things which has to be identified and they need to do the mitigation. And then analytical method validation, like we have done optimization of the method in the lab, now method should be validated. The validation of the method is such that it should be accurate, precise, linear, and then the smallest amount, that is called as the limit of detection, smallest amount can be detected. That is why the purity of the material can be determined. And then now ultimately comes to the whatever we manufactured lab, whether we have done the similar type of scale up, we need to do the characterization. So when we do the characterization, we need to do the sameness studies. Whatever the innovator material is there, our material is same or not. That we need to do as a sameness study. And that is very complex, but mainly the infrared technology, HPLC, GC, NMR, polymorphs, and solubility and particle size. So physical morphology and chemical morphology is to be done to do the sameness studies. And ultimately, the end here comes as a stability. Whether whatever I manufacture will remain stable for whatever the time it is required to be stable. Because today I manufacture, tomorrow it degrades. Doesn't mean anything to the pharma company, right? Because we are not there in the pharma company, are not there for the research, they are there for the business. So material which we design or prepare has to be stable at least for the minimum 15 to 24 months, and then more than that. Now coming to the another point is the documentation and technical submission, because as I mentioned that everything is done, but we need to go to the next level for the business, and for business starts once the regulatory gives us the approval. But that is a regulatory submission. So what we have to do in the regulatory submission is a batch record. So whatever the you prepared has to be recorded. That is recorded into the batch, called as a batch record. Then we need to compile all the R&D process development data, the quality control data, the validation data, and then all this data has to be done into the some of the standard called Alcoa standards. So it is a little advanced for you, so I'm not touching that, but it is required to be as per the Alcoa standard. Then we need to do the regulatory filing, that is called as a chemistry, manufacturing and control document. So chemistry is your process synthesis, manufacturing is how your product is manufactured, and then control is all your risk assessment. All this together is prepared one document that is called as a CMC, which is to be required to be filed in the regulatory. And then ultimately regulatory comes and audit whether you have done proper job or not. And once audit is okay, your filing is okay, you get the product approval. Now you can understand how the organic chemistry is helping pharmaceutical industries to develop and deliver to the industry and to the society. Now something which is coming, has mentioned that you also touch up with the new trends. So in the pharmaceutical industry, the new trend which is coming is a green chemistry and the sustainability. Anybody can, without reading this, anybody can understand what is meaning of green chemistry?
बॉक्स दिख रहा है सामने नीचे लास्ट में कार्टून
In
दिख
the
रहा है
hand, please.
लास्ट में नीचे ग्राउंड पे उसके बस उधर ही रखा है
No answer is wrong answer, please.
इधर
Don't
राइट साइड
consider that.
एंड
And
इधर से
then
सामने
don't
नीचे
update about your principles.
अबे इधर है इधर फोटो खींच
Very good. So green chemistry in the sense that it is not harming the earth. We call it the most, it's not harming. Like you know that organic chemistry involves huge amount of hazardous material, huge amount of the solvents, which is very difficult to recycle. So now a new trend which is coming called as the green chemistry, where we try to avoid the hazardous material, we try to avoid the solvents, by which everything is not possible, but 80% is biodegradable. And that is what the newer organic chemistry which is coming called as a green chemistry. Then another things which is coming is a biocatalysis. Like I mentioned, the biodegradable, where using the enzymes, the organic synthesis, and then the raw carbon change. And then safer solvent, like instead of, there are classification of solvent, class 1, class 2, class 3. So called as toxic solvent. Instead of toxic solvent, hazardous solvents, we call it a safer solvent. Another thing we should mention, conventional manufacturing versus the continuous manufacturing. So industries are going like, you people all are onto the Insta, you people all are into the all the social media. So my suggestion to you also look more into the, suppose you want to build your career in organic chemistry, especially in pharma, because organic chemistry can be used anywhere, like dye industries or pesticides, fertilizers, and everywhere. But if you want to go into the pharma, because I'm a pharma person, so please go and more look into the what you are looking in the pharma for the what are the newer trends. I will listed some of it here. And another technology which is called as a nanotechnology, which is coming into these are the newer subject, and then you can take the lectures on this. But nanotechnology, the chemistry is becoming more and more prominent nowadays. Now coming to the quality control and quality assurance. Like we have discussed about the research and development role of organic chemistry. Now, organic chemistry is involved in the QC and QA. Like quality assurance, you know that it is building the framework, standard and procedure to prevent the defects. Quality control is a focus on testing and 100 milligram. Let's say 1 gram. And the batch size is what? 1 ton. In 1000 kg batch, you take 100 gram. That is the highest. You never take 100 gram, but it's highest. So 100 gram sample give the assurance that my batch is okay? No, it will not. And that is where the quality assurance comes in picture. So quality control gives you that you know my testing and everything is okay. But quality assurance gives you assurance that 1 ton material which I am supplying to my indirect patient, because ultimately is going to convert to the formulation, and then ultimately it is going to be used patient, because in that case we are serving the patient. So I have to be assured that my 1 ton material is safe and effective. So QC is a mandatory requirement, QA is the regulatory requirement with the compliance, and that is ethical. It is a very simplified way of I want to just give, because we are discussing with the students. Right? So QC detect the defects. QA prevents the defects. It is product testing oriented, it is the system and process oriented. Ensure safety and efficacy of the each batch, ensure compliance with healthy regulation, and ensure that that entire batch is efficacious and safe. Outcome is verification of product quality, and QA outcome is consistency in manufacturing. So what is the role of quality control in organic synthesis into the API? As I mentioned, analytical method validation. What we do, the validation that precise, accurate, sensitive, limit of detection, linearity, all this. But there are many instruments like HPLC, JC, mass spectrometry. All these methods which require to either test or characterize the API require to be validated, and that is a regulation. Because if I don't use the validated method, I don't have the confidence whether I am testing the product is the way it should be. That is why the validation is must. And the second point, as I mentioned, for the especially for the API manufacturing in the pharma industry is impurity profile. Then now there are two things: the normal impurity, like organic impurity. More nowadays, over the years, regulation also is changing. People are learning, everybody is learning, regulators are learning, more and more new things are coming. And now two buzzwords in the pharma industry for the API is a genotoxic impurity and then nitrosamine impurity, which is a nitro group. So these are the two impurities which are the newer form of impurity which regulators are looking. Where nitrosamine impurity is it is headache for all of us into the pharma, that if the molecule itself is a nitrate or nitrite or secondary nitrite, then there is a possibility of the nitrosamine. So all these are the advanced stage, but these are the two things which we need to really look into, and we need to do when do the profiling. These are the organic impurity, related substance, inorganic impurity, nitrosamine impurity, and genotoxic impurity we need to do. As I mentioned, characterized of physical properties, which is very critical for the API, which convert to the formulation, which ultimately available to the patient into the safe and efficacious way. Stability, which is critical, as I mentioned. Then product, what we design or develop or manufacture has to be stable. And then the newer technology what called as a process monitoring, that is a PATE tool. Here, this is what happens that when we take the samples, we put into the, let's say, UD, or let's say we put into the HPLC, then they do the analysis. Now, this process analytical technology, which is a new concept, which is coming into the last 3-4 years, we are proud that we have implemented. It is giving the real-time analysis. So you don't have to prepare anything. You don't have to prepare samples. You have to just put that probe into your product or into your reactor or into your mass, it gives you the outcome of that. So these are the new advancements which is now prevailing into the pharmaceutical industries, which is new. Quality assurance like regulatory compliance, quality management systems like when we manufacture the products, many times you know that every time everything will not go okay. There will be always a deviations, there will be always a failure, there will be always a complaint, somebody mentioned that your product is not working. So all these are happening where quality assurance play a major role, equal as a quality management system. And then quality assurance, as I mentioned, that assures the quality, they will do the testing of the quality. So they are involved from the design to deliver. They are when I mentioned about supplier qualification, I always give the example about automobile. Like I always give the example about your aerospace. Like they are, I don't know how many people know that the space industry works at what sigma level. Anybody know about the sigma level? The sigma level is a process capability, like the number of defects per million. That is called as a defect level. So people are working as 6, now 8 sigma. So that is called as a process capability. So in the supplier qualification, if my supplier is not dependent, supplier is not give me the quality the way it required, because I am not going to manufacture everything. I will depend on my supplier. If the supplier is not qualified or I have not tested their capability, my ultimate product will fail. Their quality assurance will come picture, their organic synthesis knowledge will come into picture. Documentation, risk management, and audit and investigation. This is where the quality assurance role will play, and their knowledge about organic chemistry is useful into the quality assurance. And generally in pharma industry, in our API manufacturing and R&D, we generally go to the MSc and PhD into the quality assurance. I mentioned about risk management. This is a detail based. First is the risk identification. Second is the risk assessment. This is part of the QA/QC. Then risk control, risk monitoring, and risk communication and mitigation. So when we identify risk, there is a contamination, that if something not properly handled, there is a contamination one product another product. Then another assessment, there is a row. If supplier is not qualified, they give the variable product. Then ultimate your product will be finished product will be made. Then cleaning methods, then stability issues, and then other deviation corrective is not properly. So I just skip all this. Now, I have already mentioned organic impurities, residual solvents, elemental impurities, and genotoxic nitrosamine. I already discussed with you. So nitrosamine, it is very two things which is now buzzword in pharma. N-nitrosamine like NDMA, alkylating agents, epoxide, alkyl halides, sulfonates, esters. So all these are compounds which can create then when we do the sucking or staining of our drugs, this is required to be looked into to avoid such impurities. And the stability, and the stability, like there are various guidances like Q1 and Q1A to Q1M. Like stability testing of new drug applications, photo stability. Here one thing which is very interesting. So when the product is exposed to the normal environment, like some of the compounds, as you are aware about that, they are degrading into the light. So it is the mandatory requirement as a regulation. We also have to perform the photo stability. For which we have the photostability chamber, where the high level of the illumination lux levels are put and then we expose for the 3 days, 5 days, 7 days, and see them with the compound. So we simulate the product into the sunlight, like similar type of thing which is available in the photosynthesis. Now coming to, we have done the research and development, we have discussed about manufacturing, we have discussed about the QC, QA. Ultimately, what regulatory affect is because we need to file the product. So when we want to submit the product, then and then the product approval come and then business will start. So regulatory affairs encompass the chemistry, manufacturing and control strategy. Then labeling. That labeling is what? That this product require the special protection, like some product is oxygen sensitive, so you have to put a nitrogen blanket in that. To label this product is oxygen sensitive, nitrogen blanket is required. Some product is having the very high amount of the organic smell. Then we need to put bacterial carbon to absorb the smell. So all these requirement which is put into the product as a part of the labeling, and which is what is the life cycle management, compliance, operational utility, policies, and all this is a part of the regulatory affairs. So first, regulatory framework is what? That is introduction to the CMC. That is role in regulatory submission, like chemistry, manufacturing, control. Which are the guidelines is Q7, Q8, Q9, Q10. So this is ICH. Go and serve the ICH. You will know everything about the API into that and their regulatory requirements. What are the CMC components? That is a drug substance information, drug product information, manufacturing process information, and control strategy. How you are controlling your process. And specification that what you are going to test for your test product. And then analytical method validation. So these are some of the challenges. I don't want to go into that. What are the documentation required? That is a common technical document which is called as CTD. So CTD has a 5 module. Module 3 covers the CMC. Module 3 requirements include manufacturing process, analytical method, specification and stability method. Change control and life cycle during the course of your development. Anything changes which require is part of the submission. Then any post-approval changes which is required, because when we do the scale up, there are some changes required is part of the submission. And the risk, I have detailed explained about the risk management. There is ICH 29 to be part of your risk. What is the future trend in the CMC? Digitalization or obviously automation, continuous manufacturing process, and QBD. All these three are more and more required to view in the submission into the submissions. So, what is the takeaway of my presentation for the organic chemistry and like scalability is paramount. Pharmaceutical industries are there to support the patient's need, but simultaneously they are there for the business. So both the things has to be balanced. Obviously, patient safety comes in the topmost priority. Simultaneously, the business also is equally important. So, I developed in R&D, but it cannot be scaled up, no business. So, scalability is paramount. So, my process which I am developing, where my organic chemistry knowledge will come in picture to help that I develop the process which is scalable. Quality by design. I don't want to do thousands of experiments to identify which is the best. So there are the ways and means to do the identification of the which are the better way of doing the development and scalability. And regulatory alignment. I do something, but it is not aligned with the regulatory. Doesn't mean anything. Because I am not here to do the research work. I am here to do the business. So regulatory alignment is risk mitigation is critical for the business continuity and documentation, as I mentioned. Thank you very much.
Sir, API contain impurity profile. So how to separate it, and if it is there, and then how to identify it? It might be the isomer sometimes, and I think so. It is very dangerous, or and sometimes it is very important also. So
So any, answering your question, any impurity, whether isomer or organic, inorganic, or any impurity like mentioned is important. You are very, very important. Now, identification is a technique by itself. So there are various techniques in here. First thing we do the theoretical assessment. What is the impurity pathway? First in the literature, what are the possible impurities in this reaction kit? Then we do the impurity pathway. That is the second point, we get this synthesis will change this type of impurity. Third, we degrade intensely the product. We degrade the product in hydrogen peroxide, that is oxidative. We degrade the product in acid, that is acid. We degrade the product in alkaline. Then we degrade the product in the we degrade the product in alkaline. So there are very different ways to degrade the product. And then we do the analysis that what type of impurity is generated in the then we also do for the isomers and other points. We do the different temperatures to see that whatever the desired isomer is required is there or not. What is the contamination of undesired? So these are the ways by which we do, and then we finalize the process and validate it.
Yes, sir. Is there any role of other component other than API for the stability of the API component in drug molecule?
Very, very
Any
good.
linear relationship or
Excellent question. So, as I mentioned, one point, I just touched that supplier policy. As I mentioned, in the pharma, suppose it is a C1 stage synthesis, stage 1, stage 2, stage 7 stage synthesis. In pharma, we don't manufacture all the stages because it's not viable. Then we give our processes to our vendors. That stage 1 to stage 5 is required to be manufactured by them. So in that stage 1, the chemical which is used to synthesis for stage 1 is become critical. Then stage 1 itself become critical. So likewise, the supplier of the stage 1 and their chemical which is required to synthesis to do stage 1 synthesis. So that is one first point. Second point is that if my compound is very, very sensitive, my packaging material is very critical. So I have the very sensitive compound which is the oxidative degradation. Then my simple way of packing is not going to help. So I have to use a double aluminum pouch. Then I have to put the product. Then I have to flush with the nitrogen. I have to seal it. Integrated test leakage is not there. Like you see the wafers, it is there in the balloon. So we have the similar type of thing. Then we put the silica so that no moisture, no oxygen. So that is also very critical for the other than the silica. It depends on the
many years difference between the developed country and Indian country. So we are labeling British part. Difference between the quality of the developed.
I don't think that if you see, fortunately, I was part of the IP, new IP, and I was part of the new regulatory and GMP guidelines, which is substitute F, I was part of that. And I am very proud as an Indian and as a pharma professional that we are, our schedule and GMP requirement either equal or far better. Only thing, now we need to really work with all these young brains, that we need to really implement it. We are little poor in the execution. My IT, if you see, is equally as per BP, USP or anything. So I'm proud of that. Now our endeavor is a pharma professional with a young blood like this. We need to do proper execution. It is for the developed countries, no. For the other countries, but we are working like I see. We are working in some of the forums, then IT will also be acceptable. That is a big. But you know the political, you know the all these regulators, so it will happen.
Sir, this green chemistry methods you have mentioned, are these all methods are efficient for large scale production?
Excellent. So, as I mentioned, we don't do, we do the R&D, but we do the R&D to see that whether it is scalable, as I mentioned in my presentation, and is sustainable, and then it is possible. So, my experience, because I did the R&D, out of 10 compounds, my hit rate is 6. So 60% is efficient to go on. But still we are into the development stage that we want to get to it. But 60% time it is efficient.
Quality of the compound.
So what about large scale products? Are there any other alternatives? Because in photography, you will be using some of solvents. So how would you manage in large scale products? Separate out the
I am very happy to have this type of question. So, you are very right that laboratory many times when we do the scale up, whether it is sustainable or whether it is cost effective, that is the first thing which we have to do. Like in the column chromatography, you correctly mentioned about all your what we call as columns, the length of columns, width of columns, then the filling of columns, whether it is cost effective, yes or no. 99% time no. In the scale up, you have to have the different technology. In that different technology, there are always ways and means about doing the chemistry point of view, about the crystallize, how you will separate out the crystallize, the adding the anti-solvent or precipitation or the immediate cooling, super cooling, like heating. So there are many ways, but simultaneously we also need to see when we do this, your polymorph doesn't change, because polymorph is so sensitive for all such techniques. If it go development is done with the column and I'm going with the traditional chemistry, then I have to be very careful about the polymorph. So 99% time in my laboratory, I always try to do that it will be scalable to that level. I will give you the percentage, like 99.5% time, we try to avoid the column. But nowadays peptides, oligopeptides, or many of the large molecules obviously require the column, so which we cannot avoid. Like biopharmaceutical industries, like proteins, peptides, you cannot do with chemistry. You have to have the physical separation for it.
Hello. Sir, I have two questions. My first question is how nitrosamine impurities are formed and what is its use? And my second question is how do you handle batch failure?
So nitrosamine I mentioned that is based on the azido nitrite compound. So based on the structure. So it is part of that process that when we do the screening of your molecule. If the nitrite group is there, then you add something oxidate, then thrown product into that, then those type of impurities which are forming. So we need to do during that stage the killing of that impurity which is generating during that time, the azido impurities, which is the next stage for the nitroso impurity. So in that structure, if it is structure itself is having that molecule, then we need to take care during the manufacturing, especially for the oxidative type of thing to prevent those things. And then it is an inherent part of that molecule. Then the purification process is itself is become crucial. And then that purification process to control that particular impurity which is generated, we know that route of synthesis where this will be happen. So either chemistry control or engineering control which we do, and then we control that impurity. But simultaneously, as I explained to her about the packaging material, so packaging material also has become very crucial for such. So that is the first answer. Second, batch failure is always, especially for the API manufacturing, is always the nightmare because we don't know how the. Suppose I will give you one example. Some unknown impurity comes, which are not part of my specification. Then we need to do lot of user trial based on the first we need to identify impurity. We have to do the mass. Okay, which mass that impurity is. So we have to do mass spectra. Then we have to do the HPLC at what retention time which is coming. Then we identify the structure, which structure this impurity is there. Once that is the knowledge is gained, then we know that in which type of synthesis, which parameter or which environment has created this impurity. And then we go to the cause for that and then we control it. That is how the normal process is. People are trying. So many things which you have mentioned, like we are working on the flow chemistry. We are working with the single pot chemistry. So currently for the 1000 kg batch, we have 5000 liter reactor. Now this single pot reactor is just one liter. So all this water is going through that reactor is coming down. So we have tried two products and we are successful. Flow chemistry, the common which we are putting into the flow and then synthesis happens and coming. So these are the new technology. We are successful in new compounds. We are working. Please come to Baroda. I already invited you. Please come to the students to see our arrangement. You are welcome.
One message for the students if they want to make a career in pharma. One message for the students if they want to make a career in pharma.
So I don't want to push them to becoming pharma because I am a pharma people. But organic synthesis is a vast subject. And then the message from me, because last 40 years I am pharma, especially for me is my inherent satisfaction that I am doing something for the society and for the patient. So that is the personal level. And the pharma, as you know, as a career point of view, there are, as I mentioned, there are newer technologies, mind-boggling technologies which are coming. I mentioned flow reactors, single pot reactors. So all these are which is going on, and I don't think in the pesticides or petrochemicals or fertilizers, such type of innovations are happening. So we are really interested into the more into the R&D, innovation or patient care. Pharma is the job which you should look into. At least I am very happy. I am in the pharma. The
So-
same message I am giving to you.
First of all, it's very fruitful session. I have two questions. One, what role does biomarker plays in clinical phase trial success rate? And second is, as you know, there are many curious minds present here. So I want to ask you, which skills should young pharma researchers focuses on for R&D and pharma?
So I'm not expert in biomarker, but I look after the quality assurance of the clinical trials. So without the biomarkers, clinical trials is nowhere. So we need to identify which are the what you do into the biomarkers, at least for the NC molecules, for the zinc molecule is all possible. So you have to follow that. For the new drug applications, like our product is coming to the criteria what is the thing, where you have to identify which biomarker, you have to infuse into the animal, you have to generate that, you have to do, there are many things. But for the generic industry, at least biomarkers are found. So I don't. So there are many drugs that you can target drug delivery, there is liposome drug delivery, nanoparticle drug delivery, the protein bound drug delivery, that human albumin bound drug delivery. There are many drug delivery systems are there. But in that, the different disciplines are coming. For us, for the organic chemistry, like nanoparticle is one of the compounds. Like many compounds which are coming, which are the polymorph new complex compound. Like I have shown the structure of aspirin. One molecule I will show the structure. It is having the, even though it is not large molecule, but it is having the 7 to 8 different types to attach to the many molecules. Many things which are coming.